Hey everyone, this is Juan. I'm here with our second tutorial, and I wanted to dive a little bit more into prokaryotic cells or prokaryotes, because in the first tutorial what we did was we compared prokaryotes with eukaryotes. But on this one, I want to go into a couple more details about prokaryotes because I believe they're essential for classes like molecular cell biology. So it's time to start with our topic, the prokaryotes. And this type of cells actually is found mainly or exclusively, let's say, in bacteria. Probably heard about bacteria. It's nothing strange. If it probably rings a bell when I talk about E. coli or Staphylococcus, and they're present in yourself as well. Great part of your weight is actually bacteria, just out of curiosity. And they're monocellular, meaning one cell or single-celled organisms from the root mono. And a curiosity is that the largest or more complex ones or type of bacteria is actually cyanobacteria. They're present in the oceans and they're actually very important for the ecosystem. You can learn more about them in biology. This is not for human biology, but in general you can learn about. Also, another important thing is that bacteria do not have nucleus or nuclei in plural. They do have an area called nucleoid, and this is where the DNA is located. You can find a large molecule of DNA, and it's here. I can show you in this purple area. Now, going on with some basic characteristics of prokaryotic cells, I have to mention shapes, the shapes of bacteria that we see out there. And there are three basic types of shapes that I want to mention, but of course you can deal into more detail. But the first one is spherical shape, and it's more or less like a ball. This is how I draw a ball. Not very artistic, but you can get an idea. The second type is rod-shaped, a little bit like the shape that we have here of this bacterium here that you can see that I drew. Again, not very artistic. And the third type I want to mention is quite common also, is spiral-shaped. Basically, shape of a spiral, nothing too complex, just a spiral. And this is how I draw a spiral. Quite amazing, isn't it? Anyway, another thing I want to mention about these organisms, quite important, is that they can live in extreme environments, environments that you and I can definitely not live. And I'm talking about environments where the temperature is extremely high or extremely low. There actually, some of these organisms are called extremophiles, but I can write down here. So extreme temperatures and also extreme pHs, either too low or too high, where you and I cannot live with. On this quick tutorial on prokaryotes or about bacteria, I want to talk about a very important structure, which is considered extrachromosomal DNA, which does not belong to the main chromosomal DNA that codes for the main proteins. I'm talking about plasmids. They're found in prokaryotes. Probably you have heard, quite important in research. I'm going to try to draw more or less the structure of a plasmid. It's kind of circular like I'm drawing here. Double-stranded, so I'm going to draw the ladder-shaped DNA structure, double-stranded. And these structures, it's important to mention that not only they're separated from the main chromosomal DNA, but they can be replicated independently. And three main things about it is that it's double-stranded, so double-stranded DNA, two strands of DNA. The second thing is that it's a small molecule. These are small. That's why they're good for research. They're small, easy to handle, let's say. And it's circular DNA. So three main things about plasmids, the extrachromosomal DNA found in prokaryotes. I want to try to show you here in my beautiful drawing, very artistic person I am. But I'm going to try to zoom in so I can show you where you can find more or less these structures or have an idea of where you can find, because this is not the perfect drawing, but it will illustrate what a bacteria or bacterium is. So you see in the middle the nucleoid, the area where you find the chromosomal DNA. And then you have these structures that I'm gonna draw in green here. I'm just gonna highlight them in green here. 1, 2, and 3. These are my plasmids. To end this tutorial, very brief tutorial on prokaryotes or prokaryotic cells, also known as bacteria, I want to discuss the main structures of these organisms. Just the main structure so you can get an idea of what you can find here. And the first one is the peptidoglycan cell wall that you find, the outermost layer here that I'm going to highlight. So actually peptidoglycan means that it's comprised of polysaccharides and peptides, and I'm just outlining it here. The innermost layer is actually a cell membrane, a lipoprotein cell membrane, comprised of phospholipid bilayer associated to proteins. And I mentioned that in the first tutorial when I compare prokaryotes versus eukaryotes. And here is the innermost layer. The third structure that you see not only in prokaryotes but also in eukaryotes, these are the ribosomes. And you do need these. These are quite important because these are the structures that you will learn later on that help you go from DNA to protein. The fourth structure that I want to talk about is genomic DNA. And the genomic DNA is found in this area here that I'm going to highlight, this purple area here. This is the nucleoid where you find the chromosomal DNA, or the DNA that encodes for the most important proteins of the cell. So nucleoid region. And also, let's not forget about the plasmids. These are structures that carry some DNA as well, and that I mentioned before. And now the structures that I want to talk about very quickly is that in prokaryotes you find free enzymes. I just highlighted them there, free enzymes, and also respiratory enzymes. These are quite common in these organisms. So now you have an idea of the main structures that you find in bacteria or in prokaryotic cells.